Okay, so welcome to this next video in which we're discussing the process of hemostasis. Okay, so we've made a small cut in the side of our arteriole, and that's allowing blood to move out of the arteriole. Okay, so we're now discussing how do you fill in that cut, which is allowing blood to leave the lumen of the arteriole and seep out. Okay, so this is the process of hemostasis, and I've told you that the platelets, or thrombocytes as they're also called, are extremely important in this process. So within the blood, another constituent of the blood is that you have these platelets or thrombocytes, which are small sort of fragments of cells. They do not have nuclei. Instead, they are basically made from a much bigger cell, okay, in the bone marrow, which breaks little fragments of its cytoplasm off, and those become the platelets. So what will happen is a little sort of fragment will break off, and this is what will become a platelet. And this sort of cell that is producing platelets like this is known as a megakaryocyte. Okay, and they are present within the bone marrow. So, ooh dear, is it going to fit on? Just, there we go, megakaryocyte. Okay, and it's producing these thrombocytes or platelets here. Right, which are going into the bloodstream. Okay, so how are platelets going to be important in this process of hemostasis? Well, basically, they are going to aggregate together and make a plug in the cut, basically, to stop the movement of contents outwards. So let's discuss this activation process by which we can activate the platelets to actually aggregate in this way. Okay, so let's redraw our blood vessel, but now with a cut down the middle of it, because this is going to be how the platelets are going to be activated. They're going to sense the collagen and also other proteins that they wouldn't usually be exposed to before the cut has happened. Okay, so basically, initially what you had were these endothelial cells which were lining the lumen of the blood vessel. So all the platelets saw were the luminal faces of these endothelial cells. Now we've got this cut. Instead what we'll have is like this. So you'll have, here are these endothelial cells here. Okay, like so. And here's the cut up here. So let me just finish this picture off. So these are endothelial cells. This is the cut up here. The basement membrane has now also been cut through. So you've got the basement membrane here around the other endothelial cells, but not now at the place where you've made that cut. So you've cut through the basement membrane over there. Okay, so here's the basement membrane around the rest though. And then around that you'll then have what's left of the tunica media. So I'll show the tunica media here. Okay, and then also the tunica adventitia. So you've made this nice cut through the middle here. Okay, and then you'll have the tunica adventitia, which I'll just draw as a yellow line in order to avoid having to colour in a great big segment again. Okay, so here's the tunica adventitia in yellow here. Okay, right. So, what's going to happen now is that these platelets are going to be exposed to some very different stuff than they would usually be exposed to. So, let's start with von Willebrand factor. Okay, so for this we need to talk about the way that the endothelial cells are arranged sitting on this basement membrane. So basically, usually what you have is, if I draw a bigger picture here, if this is the basement membrane here, so let's have the basement membrane here in turquoise, and then let's have endothelial cells sitting upon it. So here are the endothelial cells. Then what you have are very tight junctions between the endothelial cells, like so. And then here's the next endothelial cell along. Okay, you'll have one of these tight junctions here. So these are actually called tight junctions. Now let me explain what these tight junctions do. Okay, so they connect the neighbouring endothelial cells, and basically they separate the endothelial cells into two portions. They separate them into an apical portion, so this in orange, this is the apical face of this endothelial cell, and then they also produce a basolateral region, which is in pink here. So this is the basolateral portion. Okay, so there's two portions of the cell membrane basically, a basolateral portion and an apical portion. So in orange, this is the apical portion of the cell membrane of the endothelial cell, and then in pink here, this is the basolateral portion. Now, these two are kept distinct basically, so things which are in one of these membranes, such as the apical membrane, are not allowed to go into the basolateral membrane. So say if you've got proteins in the apical membrane, they're not just free to move into the basolateral membrane. And to make this more obvious, it would probably help if I drew a picture of this looking from this aspect rather than from the side. So if we look from below, as far as this picture is concerned, then what we'll see is these sort of cells like this. You'll see these cells which are often compared to fried eggs. So you've got the nucleus at the centre here, and then the sort of yolk, the white spreading out. Okay, and then you'll have a neighbouring endothelial cell here. So here's the neighbouring endothelial cell again with its nucleus here. And then here's another endothelial cell. So this is a picture where we're looking from this aspect now. So we're seeing the endothelial cells from above. And then what you have, basically, is the tight junctions. Although here they just looked like a little line connecting these two, in actual fact, what they are doing is they're spreading out between everywhere. All of this sort of space in between endothelial cells, this is where the tight junction is. So it's this two-dimensional structure rather than this one-dimensional structure, and when we took this cross section we only saw that portion there. Okay, right, so this is often compared to basically if you've ever bought a six-pack of beer. So if you buy beer in cans in the shop, so let's say here is a beer can here. Okay, they often come in six-packs, so I'll draw this here. So you have these beer cans here. And in fact, I'll just draw a 4-pack. So here's another 2 beer cans here. One there, one there. Okay, now, basically the beer is, instead of putting it in a box, instead the beer cans are free, but they have this plastic coat sort of put around them. So they have a sort of two-dimensional plate of plastic which is intertwining around the beer can, so it will go around the beer can. So imagine a piece of paper made of plastic, and then with holes in the piece of paper the piece of plastic where the beer cans are going to go through. That's kind of what these tight junctions are like. They're similar to this piece of plastic. They completely surround the adjacent cells and separate them into two portions, an apical portion which faces the lumen of the blood vessel and the basolateral portion which is below this tight junction, so similar to the portion of the beer can below this piece of plastic. Okay, now this means that if a protein is in the basolateral portion of the membrane, it cannot just diffuse into the apical portion of the membrane because it can't get past these tight junctions here, so it will meet a blockade once it gets here. Basically, it won't be able to get into the apical portion. So this means that you can put proteins in the basolateral portion of these epithelial cells and be confident that they're not going to end up in the apical portion, so they're not going to end up exposed to the blood. Now, one such protein which you do this with, and it's present in the basolateral membrane, so I'll draw it just as this little line here, so it's in the basolateral membrane and not exposed to the lumen. Okay, so the usual constituents of the blood cannot see this protein because it's in the basolateral membrane of the endothelial cells, so it's not exposed to them. And this protein is known as von Willebrand factor, okay, or VWF for short, von Willebrand factor, okay, and this protein is one of the key components of of the hemostasis process. Oh dear, what have I done? VMF. No, that's not correct. VWF. Von Willebrand factor for short. Okay, so we've got this von Willebrand factor in the basolateral membrane of our endothelial cells. So what? So what does this mean? Well, basically, when you make this cut, what's going to happen? The tight junctions are going to be disturbed, basically. And what this may well mean is that the constituents of the blood become exposed to this von Willebrand factor, basically. So if you imagine making a cut, okay, where you cut across either the tight junction or you're going to cut across the endothelial cells. So let's say our knife has come in and we've cut through here, then what's going to happen? This side of this cell is going to become cut away from this side of the cell. So you can imagine these two things are going to come away from the basement membrane. So you'll have this gap here. You'll have a means by which the blood can gain access to the basolateral aspect of these endothelial cells. So let me draw this. Okay, so let's say we've made a cut down there. Okay, so here's now the basement membrane cut in half. And now, what we've got is here's this old endothelial cell here. And then we've got this half of the cell left here. Okay, and then we've got this other half of the cell left here. And then a tight junction still existing between there. But now what you can see is that the blood has a means by which it can gain access to the basolateral portion. So you've disturbed the tight junctions, and it means that now the blood can gain access to the basolateral portions of these endothelial cells. Okay, and this is going to start off the adhesion process of the platelets to the wall of the blood vessel where the blood vessel has been cut, basically. Okay, so what's going to happen is the platelets have a protein on their cell surface membrane which can bind with von Willebrand factor. Okay, so let's show this. So if we have our platelet here, so this is our platelet, this little blob here represents our platelet, and then we'll have some protein on the surface of this platelet. So let's have a protein here on the surface of our platelet, drawn quite small, but never mind. Okay, so this protein is by the name of glycoprotein 1b95. Okay, so this has the rather fantastic name, glycoprotein, which basically means a protein with some sugar groups stuck onto it. Glycoprotein 1b, and then we have 9 in Roman numerals, of course, because it's grandiose. Okay, and then we've got 5 here. So glycoprotein 1b95 is the name for this protein, and it's often abbreviated just to GP1b95, or you may well just hear people abbreviate it to GP1b. Okay, but strictly speaking, you should say GP1b95. Okay, so what does this protein do? Well, its role is to bind to von Willebrand factor. So basically, when the blood constituents now have access to the basolateral side of the endothelial cells, what will happen is this GP1b95 will bind to the von Willebrand factor that's now exposed to it. So let's show this happening. So where should we show this happening? We'll draw another little picture here. So well, yes, I'll draw another picture on the other side. Okay, so we've got our basement membrane here. So let's draw it bigger this time. So here's our basement membrane. Okay, here are our endothelial cells, and let's just draw the one that's been severed now. So here's the one that's been severed. So it's got a cut down here, and then here's the other side of this endothelial cell, and then there would have been tight junctions between this and the next endothelial cell along. Okay, right, now what's happened is we've got our von Willebrand factor now exposed out here. So maybe you'll have some von Willebrand factor here, because if the membrane of this severed portion reforms, it will now be continuous. So all of the von Willebrand factor that was on this basement portion can come round into this apical portion here. So let's say this is now our von Willebrand factor in red here. Okay, so this is von Willebrand factor. I'll just label it up. Von Willebrand factor. Okay, and it's produced by all endothelial cells within the body. Von Willebrand factor. VWF for short. VWF. Okay, now what's going to happen is that our platelet is going to come in here. Okay, and this is out of proportion, of course, but here is our platelet. And what has it got on its surface? Well, it's got glycoprotein 1b95 on its surface, and these two are going to bind to one another. So this is glycoprotein 1b9, like so, and then 5. Got to get the Roman numerals looking nice. Right, okay, so I'll colour this in blue as the colour we were colouring it in. Okay, so here is glycoprotein 1b95. Okay, on our platelet. So this initial process of a platelet sticking to the exposed von Willebrand factor on the endothelial cells is known as platelet adhesion. So the first process you get is platelets adhering to the exposed von Willebrand factor on these damaged endothelial cells. So this is platelet adhesion. Okay, now this has not yet formed a hemostatic plug, so let's go on with the process now. So the next process you need it to happen is you need these platelets to activate, basically. Okay, and in order for platelet activation to occur, the platelets need to be exposed to usually collagen. And we'll continue this discussion in the next video.